Biomedical engineering is interdisciplinary, bringing together multiple fields, perspectives, and technologies to come to bear on the problems facing medicine today. The International Genetically Engineered Machines Competition, or iGEM, does exactly the same. Founded in 2003, it's a program for undergraduate and high school student teams around the world to come together, chase new ideas, explore new possibilities, and push the field of synthetic biology forward. Coming off their gold medal performance at 2021's Giant Jamboree competition, and having both occupied the executive director chair of UBC's iGEM team, Emilia Chen and Janella Schwab sat down with me to talk about their experience, how it inspired them to study biomedical engineering, and where they hope to go next because of it. Welcome to SBME Interfaces. So today it is my pleasure to interface with Emilia Chen and Janella Schwab, both representatives of UBC iGEM. Emilia is a fifth-year student studying microbiology and immunology and computer science. She is the previous executive director of UBC iGEM, having previously served as software lead and wet lab member as well. She is passionate about the intersection of computational and biological sciences and about interdisciplinary collaboration in general, which is something I'm very passionate about, same with SBME. And her iGEM experience has truly transformed her undergraduate path, which is something I really want to talk about later. Janella is a fourth-year honors student studying applied animal biology. She's the current executive director of UBC iGEM, and she hopes to inspire admiration for biological systems in others, especially those outside the scientific community. She is particularly interested by the parallels between natural and human worlds and how synthetic biology can narrow that gap, which is a very interesting thing to say. We can dig into that later too. And in her spare time, you can either find her climbing a mountain or at least thinking about mountains. Welcome, Emilia and Janella.
Thank you for having us, Miguel.
Yeah, thank you.
So Emilia, we'll start with you. Can you talk just a little bit about your experience with iGEM, the UBC team, and why you think it's a really special opportunity for students?
Yeah, for sure. So I have been involved with the team for the past 3 years and moving on to the fourth years. So I joined as a second year, and then back then I had no research experience. I was just doing my combined major in microbiology and computer science. I was looking for opportunities where I can actually integrate the two together, because in my classes, both of them are pretty parallel. I'm just learning both knowledge. And then I found UBC iGEM, where they integrate multidisciplinary research together, and it's a student team. So I joined as a wet lab member first, and then I learned a lot about synthetic biology that actually started to get me thinking about designing things, like in the designer mindset. I can create things, I can engineer biological systems for things that we can use in the real life to address problems. So that was very exciting to me, and that was nothing that I have experienced in class before. That was why I decided to stay for the second year. And second year, because I have some computational background, and I also learned a bit about mathematical modeling while in my first year time at team. So I decided to take up the role as a dry lab lead. And it was a bit interesting that in the second year we had COVID. So we actually didn't have the opportunity to go into the lab, and we took up a completely computational project. In my experience, it was really exciting to me, particularly because we did more about how machine learning can be applied to address biological problems. It's actually what I was excited about, and that aligns with my other courses very well. That year, even though we didn't get into the lab, that was a great learning experience for me, for the team, to learn more about how software, how computational technologies can be used to solve biological problems. Actually, on the team, not all of us have experience with machine learning. I'm sure Janella also learned a lot during that time about how machine learning, deep learning things work, and that was truly transforming. And then that also inspired my more of a career choice as well, to go further into computational biology graduate studies. So and then moving on to that third year, I really want to just get more involved with the team, and then I took up the position of the executive director. So in that third year, I feel like I learned more about the administrative stuff, how like how managing a project is actually like, how to balance wet lab, dry lab, and how to integrate different sections together. And that's also nothing that I've experienced in class, and that also prepared me better to be to do more of a postgraduate research. And in the future, when maybe I need to do my own project, I kind of know what are the wet lab components, what are the dry lab components, and as well as many other external things that related to research is like, I think one of the interesting, very great things about iGEM is we not only deal with the research, we also deal with how to communicate this research to the outside world, how to link our product to application in the real life, how to talk to people. And then, so I think that aspect is also very unique to iGEM.
Absolutely. And Janella, I know that you had the opportunity to participate in iGEM as a high school student. Can you talk a little bit about how that came about for you?
Yeah, of course. So just to clarify, iGEM is a competition that spans across 46 different countries. It was initially founded at MIT in 2003, and it's an annual competition where every student team that can be student-led and student-generated team creates an annual project to present at the international competition at the end of the year around October or November. So it really brings all these students from interdisciplinary fields. As Emilia said, you could unexpectedly need some dry lab initiatives in addition to your wet lab ones. So you bring about more computer scientists or engineers, things like that. So that is something that appealed to me in high school, as you mentioned. So I participated in iGEM as a high school student, since there are not only university teams, but also high school teams. And what I thought was so great about it was that in that moment, I could really see what the future was like, or it was my first experience with being in a kind of field where I really said like, wow, this is what the future is made of, like creating all these novel engineering initiatives around organisms that already exist instead of creating new products out of scratch through manufacturing initiatives. I thought that was something really great, like being able to create new products with what already exists in nature. And I went to high school in Peru. So what particularly drew about it to me was that in a third world country where sometimes science is very inaccessible, especially for initiatives around new and emerging fields like synthetic biology, iGEM was something that really increased the accessibility of science to me and my school and my team. And I think it's something that it does for all the different teams from around the world. So iGEM distributes DNA kits to every team to carry out their DNA-based synthetic biology projects during the summer. So every team has immediate access to the resources they would need to pursue a project like this. So at least personally, back home, I'd be like, I really want to get my hands on things around biological innovation, but there would really be no channel for me to do that because of resource limitations. Even in our lab in my school, which was one of the most advanced schools, there was not many resources, but iGEM allowed me to do that. So that's something that really appealed to me as a high school student, and something that encouraged me to continue being part of iGEM at UBC.
That is really, really cool. One of the things I really love about what both of you have said is it's one of the important things we're trying to get across at the School of Biomedical Engineering too, which is that education has to be a wholesale thing where you have access to these new technologies, these new opportunities, the ability to ask these new questions, but more importantly, leadership in training in leadership, right? Like you are literally leading a team and leading a project, and it's very like leading your own lab in the future. This is very fascinating. Now, speaking about the team, your team won a gold medal at the iGEM Giant Jamboree competition in November, and you were also a runner-up, I think, for Best Diagnostics Award, right? Is that correct?
Yes, we were
Okay.
nominated as one of the best diagnostic.
Good. Okay, fantastic. Can you tell me a little bit about that competition, what it's like, what you have to do to get there? And we'll start with Janella.
Yeah, so usually on normal non-COVID years, during the year, we begin our project cycle around December. So after the competition ends in November, and we begin with project ideation. And I think one of the great things about iGEM, which is something you mentioned about us, about the parallels between leading something like iGEM, and for example, leading a lab, obviously on a much smaller scale, is that you can take it really wherever you want. Just like when you open a lab, you can do it in any field you want, clearly dependent on the previous experience you have. In iGEM, you can make your project address any field. So even fields that are seemingly not touched by biology, like for example, manufacturing and things like that, you can take your project there during the project ideation stage. So our team has previously done medicine-based projects for the past 3 years, I believe, but also some agricultural ones, food and nutrition-based, I think a manufacturing-based one, I believe. So this project ideation stage and kind of consolidating what field our problem to be addressed in is in goes up until about March or February. And then after that, all our different sub-teams, including wet lab, but also maybe less thought of sub-teams like dry lab, where we do modeling and software initiatives to complement any, for example, research limitations we would have in wet lab. And also human practices, where we try to determine the social and ethical impacts of a project. That goes on from March basically until September, with the bulk of the wet lab work being done during summer in our lab at the Life Science Institute. And then September and October are, I almost said plagued, but that's too negative, but bombarded with all these competition deadlines, where we kind of consolidated all the work we've done for the competition. So that's kind of what a usual year looks like in getting to the competition, which is what you mentioned. And this past year, the competition was online, so that was a whole different experience for us. But usually it's in person in Boston, which is really exciting because we get to meet all these different teams. There's about 350 every year. It started out with about 4 teams in 2003, and every year it increases exponentially. This year, they've announced that it is going to be in person again, this time in Paris, which is really exciting.
That is really exciting too. So, and Emilia, you can speak to this a little bit. So what is the actual competition phase like? Like, what is it that you have to do? How do you present? How much time do you get to present? That sort of thing.
Yeah, for sure. So the actual or the conference, the competition is year long, but the conference is happening at the end of the competition. It's usually for non-COVID year, it's around 4 days event in Boston. But this year, last year, it was online, so they extended the period to be around a week or two. And then so we have judging sessions where we have a 20-minute presentations to talk about what is our project, what is the components within the project, and what we've done in the lab, what we have done for the mathematical modeling to model how our biological system works, and also talk about how we think this product will be used in the real life. And then after that 20-minute presentation, we'll have around 20 minutes of Q&A with a panel of judges who are in different fields of synthetic biology to ask our questions, and then just for clarification. So that is the biggest component. And then the second biggest one is for the project wiki. So every team will make a web page about their own project. So the deadline is prior to our actual presentation at the end. So the judges will first look up our website to learn about our project, and then listen to our presentation and then ask questions.
That's fantastic.
And then for usual year, we actually still also need to make a poster. So there's a poster session where all the teams just booth at the conference center, and then people can just come visit, judges come visit and interact with you one-on-one. But this year, because everything's online, so we didn't have to make a poster.
Mm-hmm. Now, so for everybody out there, can you, Janella, explain to us the project? Because I think it's utterly fascinating, which is effectively, if I remember correctly, using Salmonella as a biosensor. Is that right? Okay,
yes.
Take it away. Yeah.
So our 2021 project from last year, as I mentioned, our projects have been kind of accidentally medicine focused for the past few years. So last year we wanted to address issues of diagnosing or monitoring tumors in response and their response to immunotherapy. So usually the goal of immunotherapy is to be able to maximize the body's immune response against tumors. And then after that treatment, the usual way that clinicians see if it's being effective or not is a tissue biopsy, which can be invasive and uncomfortable. especially if having to be done repetitive for patients. So we thought of making a internal biosensor that kind of lives or is already in the tumor that can release these continuous outputs of how much immune activity there is in the tumor after an immune treatment. So we took a kind of unconventional or interesting approach of using naturally tumor colonizing Salmonella, which is non-pathogenic strain and has actually been used as, is actually starting to be used as a cancer colonizing agent in clinical trials. So we know this is not a really random idea of putting bacteria inside a patient. And then through this ability of it to naturally kind of live in the tumor's microenvironment where the tumor interacts with the body's immune cells and fibroblasts and blood vessels and things like that, be able to excrete these outputs of how much immune activity there is. So we did that by having reporter molecules whose production is induced by the presence of certain immune markers that indicate anti-tumor activity inside the tumor microenvironment. So this was actually a bit difficult, and the choice of molecules to indicate that was a bit challenging, and we talked to a lot of experts to get their opinions on this, and we ended up choosing response to TNF alpha and lactate as proof of concept, so that the presence of these two molecules, and only these two instead of one or the other, would be needed to produce our reporter molecule, which would be then collected in urine. So this is the project that we developed last year. It was lots of fun since we hadn't worked with Salmonella before. So we got to talk to some MSL faculty to be able to give us some Salmonella strains and learn cell culture and inserting DNA into it and all that fun stuff. So I think overall, it was a really fun project, and it did take us to being nominated for Best Diagnostics Project. So that was really rewarding and fun.
That's so cool. And Emilia, I'd love to get your opinion on this. You've been in this now for 3 years, as you say. And she just talked about working with these experts, right, in differing fields. What is that like, getting that kind of insight and that kind of exposure to people who are working, at the leading edge of their field?
It's actually so surprising. And I felt, so first of all, as an undergrad, I actually didn't expect that I would have the opportunity to interface with people leading this field, like top scientists, to learn about their insight, learn about what they think about our project. So it was so exciting that we can get response. So we typically would send out emails to give them a brief introduction of our project, and then ask for like requests of meeting, or we have specific questions for them. And then we always be so surprising to be able to talk to them. And during our chat with them, it was also really eye-opening to see what kind of cutting edge technology is, and to appreciate their generosity. So actually, when we talked to some experts, they were like, oh, we actually have the cell line growing in our lab. So if you do need it in the future, maybe we can collaborate or things like that. So that was actually very exciting to be able to interface with top researchers.
They're always really generous, or surprisingly generous, especially as undergrads, where before any research initiatives, your interaction with professors is usually through classes or in labs where they already have a project for you. It kind of, you don't really realize it until you're in an initiative that like this, that your ideas can be really valuable even to professors, to the point where it entices them into providing resources for your idea, which is really nice.
Now, I mentioned this a little bit in the intro, especially for Emilia, but I want to get both of your perspectives on this. Is you've both talked about how iGEM exposing you to synthetic biology it's sort of changed your direction, we'll say, through your undergraduate degree. And so I'd love to hear about both of your experiences with that, about how this exposure has now changed the way you want to go in the future. And we can start with Janella.
Yeah, so ever since I had that exposure to synthetic biology in high school, I remember the moment where I was at the in-person Jamboree in Boston, just walking around, looking at all the different projects that really have potential in the end to be, for example, commercializable. Like you have this bacteria that can produce in our case, obviously, for last year's project, there would be a few, it would take a long time to get there, as would be for a lot of other iGEM projects. But in projects like that, you'd be like, wow, this is actually a system like this Salmonella that you could put inside a drug and administer to patients. Like, this is actually so commercializable. This, more than an educational initiative, which is the main goal, it also can result in really cool commercializable and very commercializable and viable products that can be used to further expand the biotechnology industry. So I remember going around looking at all these different projects with a lot of potential and thinking like, wow, I think this is, just by myself, without having had any previous exposure, extensive exposure to what the general public feels like about synthetic biology, I remember thinking like, this is the field of the future, I think. So I remember keeping that emotion and keeping that thought throughout high school, and then coming to university, and then not really knowing how to get involved with that again after that experience in high school, since back home in the rest of in my subsequent years in high school and first year of university, it was kind of this abstract thought, because it was such a new emerging field that I didn't really have that much time, like that many resources to know how to explore it further. Then when I arrived at university, I actually wanted to pursue veterinary school. So I had thought that synthetic biology was a great experience, but I didn't really know how to really get my hands on it on a professional level, or if there was even a professional viability for it at the moment, or it was just too new. So when I joined university, I actually wanted to go on a veterinary path, but then, and that's why I'm in the applied animal biology program. But then after joining iGEM and having such an extensive exposure to it, and through it being able to meet professionals in both academia and industry that are working on real tangible initiatives to advance synthetic biology and commercialize it and really make it affect every industry, like manufacturing or food systems and medicine and things like that, where I realized that this would be a viable future career path for me to pursue. And ultimately that's led me to join more concrete synthetic biology initiatives. For example, I was a co-student at Nika Shakiba's lab, where I worked on synthetic biology projects. And I'm now working on a thesis with her, also centered around synthetic biology, and I do plan on pursuing it in my future as well, at least at the moment. So that's a bit of the journey in my head about how iGEM has kind of derailed my whole career path. Derailed is maybe a negative word, but derailed into a good outcome.
Yeah, no, I like that. And Emilia, what about you? How was yours derailed?
I wouldn't say it's derailed. I think it's like making it more concrete and making me to realize what field actually excites me. So although I started with a combined major of microbiology and computer science, I didn't have any idea on actually how to combine the two. I just thought, oh, maybe it's cool experience to have two majors together, but I didn't know what's the use of it. So in seeking more experience of this, I joined iGEM. So that was the first time I truly see like interdisciplinary research happening together. Although as a student team, we do have different aspects in experimental part, engineering part, how math and computer science contribute, like how they work systematically and integratively to create a viable product at the end. So that was a very appealing experience. And also through iGEM, I was able to meet a lot of people with a similar mindset with interdisciplinary studies. So I said that I was in a combined major. This combined major field is actually pretty small at UBC, but at iGEM, I do find it in reach of people doing combined major in both biology side and computer science side. I also was able to talk to more experts in the field that have this combined experience, especially in our second year when we do a more of a computational focused project. We actually interface with the founder of UBC iGEM, who has both computational and biological background as well. So that was very exciting to me. And that also encouraged me to look further into how I can actually use the two together. And then I think we mentioned previously that at iGEM, we do have a variety of different research topics, from medicine to agriculture, food and nutrition. So by looking into different projects and having experience in multiple things, I kind of know that in the future, I do want to go into more medicine-focused research for graduate school. And that also encouraged me to get more involved with SBME research. So right now I'm a direct study student at Carl de Boer's lab and also doing interdisciplinary research with computer science and biological engineering. So that's also very exciting. Like
This is
that's
awesome.
how BioGem has been narrowing down or making my future more concrete to me.
Yeah, that's brilliant. And I now realize that you are next door to each other in terms of lab space, right?
Yeah.
With Nika Shakiba and Carl de Boer. That's excellent. Okay, so then this is for the both of you. Imagine I am either a high school student or first year undergrad. I'm interested in taking part in iGEM or maybe learning toward the biomedical engineering side of things. Based on your experience, what advice would you give me? And we'll start with Janella.
Do you mean high school and first year because you mean more like starting university path?
Yeah.
Yeah. I think first of all, there's no limit in university to what kind of or what year level of students we take on. So we have had multiple first year students join our team, and those are often the most enthusiastic and long-lasting team members we have, because they feel especially special being granted the opportunity to join us, and they often stay for the entirety of their undergraduate degree. So first of all, in that jump from having no experience in this field in first year to being an iGEM, that jump is not that large. Like, we take first year students who have had very limited experience in synthetic biology or in the wet lab. But then in steps before that, I'd say there's many resources that are out there to learn more about synthetic biology. I think iGEM does a really good job of that. It often has very public and informative resources on, for example, safety of synthetic biology or public policy and things like that. And those resources are open for anyone. And even the attendance of the competition is free for anyone. So I know one of our members who's a second year now and joined as a first year, she attended iGEM as a high school student, just attended the Jamboree just because she was interested in the field. And that is a way to learn a bit more and get a bit more involved in what iGEM does and see if it is something for you. So that's also something I really like about iGEM, that they're really good with kind of educating people about synthetic biology, especially because it's such a specialized and new field, which makes it a bit vague. Even if you look on Wikipedia, the definition is like, there is no set definition of synthetic biology. It's kind of just a merging of biological engineering, systems biology, utilizes genetic engineering. So we don't really, or iGEM knows that they don't really expect people to know exactly what it is. So they do a really good job of educating people about it. So I'd say go out there and learn things through the resources they offer. Starting out with very advanced research, like for example, papers or things like that, can be a bit daunting. But I think iGEM as a foundation and also previous iGEM projects are really good at explaining things for younger people.
And Emilia, what would you tell me?
Yeah, so I think first I want to build up on Janella's point of reading up like other things that iGEM has provided. They are actually facilitating open communication to outside world about synthetic biology. So there are also a lot of web page on previous iGEM projects, then you can click into it. And also they are trying to make it more accessible to people. So we are starting to make videos about synthetic biology research, and then some introductory videos from iGEM are very good to get to know more about it. And then I think I would say second thing is don't be afraid to reach out. So we actually had a really enthusiastic high school students reaching out to us, trying to get to know what we are doing, try to see whether she can join our meetings and see how we are organizing things. So I believe we're still in touch with her. And then she said, oh, maybe when she comes to UBC, she will also come to join our team. So just don't be afraid to reach out and then to talk to people, learn more about this initiatives. I think that's what I would say.
I love that. And I'm very cognizant of your time. So we only got a couple questions left here, I promise you. But no, I love this because it's really inspiring, I think, too. One of the other big things about SBME in particular is we're trying to be representative as best we possibly can, right? So that somebody can look at a biomedical engineer and think, there, I see myself there, right? And part of that is part of this trying to redefine that vague definition, right? Like trying to, for the longest time, people thought biomedical engineering was the person who fixes the broken MRI machine at the hospital, right? And it's so much more than that, right? So yeah, this is wonderful to hear from me because it means that message is getting out there one way or another. So I love that. So, okay, so next question then is actually sort of in the realm of the last one. If you could give yourself advice at, say, 15 to 16 years old, you talk to your 15 to 16-year-old self, what would you say? Emilia, we'll start with you.
Wow, that is a good question. So I think 15 and 16 year, I was in high school. I actually, so I grew up in China and then I came to Canada for undergrad. So I guess one thing that I would tell my 15-year-old self was to get broader interest in more than just studying. So actually in my high school, I just focused on like more like physics, biology, chemistry, that kind of disciplines, but I never really touched computer science. I also never really touched like art design thing, which turns out to be very important in my current life and also my experience at iGEM. So for example, at iGEM, we do interdisciplinary studies. So that's why computer science come into play. And we also need to graph up our projects so that to facilitate this scientific communication. That was also something surprising at iGEM is how much design work that we have to do, like graphic design work, how we make how we make slides and to showcase our project. So I think I will give my 15 and 16-year-old self to just explore more about your interests, explore things outside of studies, explore things that you've never really touched in school. And don't think that is like something against like the common situation. It's just to get to know more things. Yeah.
I love that. And you, Janella?
Before I go into that question, I just like to touch on what you said about kind of how SBME is trying to redefine what biomedical engineering is and kind of representation of not only different kinds of people, but also different kinds of initiatives. So I think something that's great about iGEM and synthetic biology is that it's kind of blurring the line or extending, I guess, the realm of what we can engineer. So I agree with you that at least before I interacted with any SBME faculty on any degree, I thought, oh, biomedical engineering is kind of like medical devices, things like that. But recently, SBME has definitely been getting more interest in cellular engineering, and it's really blurring the line of what is engineerable or codable, because synthetic biology at the end of the day is like coding life itself. I mean, I think in not so recent years, but recently the ability to program computers and things like that has been a huge, clearly because we are on Zoom right now, a huge breakthrough, and continues to be even more emerging and extending the line of that more. But I think now we're realizing that we can program different organisms to be able to result in the same outputs we would want from, for example, a computer. And I think initiatives like SBME or like faculties like SBME are realizing things like that. And I think it's really great that they're making active efforts to kind of expand or rebrand what it means to be a biomedical engineer through having a lot of synthetic biology based labs and synthetic biology based or cellular engineering based initiatives. I think that's something that's really great. And I also think it's kind of becoming, or at least the way I see it, it's kind of becoming a commonality between different life sciences fields that are often not connected. So for example, the first thing we're ever taught in biology is like, oh, animal cells, plant cells, and bacteria, they're all so different. There's nothing between them that can be translatable almost. They're completely different systems. But now with synthetic biology, we're trying to standardize the ability to engineer all these different systems. So we can have plasmids, which are building blocks for synthetic biology and cellular engineering, that are translatable between bacteria, different strains of bacteria, between bacteria and plants, between bacteria and mammalian cells and animals. So I think it's kind of becoming this common theme between all life sciences fields that, for example, you wouldn't think of if you were only in one. Like, for example, in biomedical engineering, even if you were in cellular engineering, maybe you'd be like, oh, there can be no commonality between this and things like bacteria or plant systems. But for example, through projects like ours last year, we can see how the worlds of microbial Salmonella world and very medical engineering-y things like immunotherapy and things like that can be brought together. So I think that's something that's really nice and becoming more of a standardized commonality between different life sciences systems. That was a little rant on the side, but your question about
that was a great rant.
Yeah, I just wanted to touch upon your point about SBME before I forgot it, but your point about what I would tell my 15 or 16-year-old self. I think at that point, I was also in high school. I did have an interest for biology primarily, but I was trying to explore many different things at once, or trying to explore what it was I liked the most, or I wasn't sure what I liked the most. So for example, I'd be really interested in I thought like conservation and ecology was really cool, but then I got to volunteer at a few vet clinics and I was like, ooh, veterinary medicine sounds really fun. Like I like animals, that makes sense. But then I feel like I tell myself to hold on to what was truly more than what was tangible at the time. Like, for example, around me, I could see a lot of people pursuing veterinary medicine. So I'd be like, oh, that's what I should pursue because it's just very easily accessible. Like, I know what path I need to get there. But instead of going through that route, I kind of tell myself to go with what sparks the most passion or curiosity in me, which was kind of the things I did at iGEM and the experience I had at the Jamboree. That was one of the first moments where I was truly able to see or truly able to feel or be touched by what can be done through science and the merging of science and engineering. But I think I didn't hold on to it because I didn't really know how to pursue it, or I didn't make it. Yeah, I didn't pursue it further because I couldn't see anything around me that was a tangible path forwards. But I would have told myself to kind of try to explore it more, like use more the resources I had available. Like, for example, the people I met at the Jamboree or my my previous mentor, the one I had during high school, who led iGEM, things like that. I think that's what I tell myself.
That's excellent advice. I mean that like wholeheartedly. Anybody I speak to at that age, the first thing I say is go do things that interest you, period. Like that's it. And then and try to figure out why they interest you while you're doing them. This has been fantastic. Great conversation so far. Okay, so and then last question for the both of you. Are there any particular initiatives or projects or endeavors that you are a part of right now that you're excited about and you feel that we should get excited about too? And Emilia, we'll start with you.
Some kind of initiatives at iGEM or anything?
Anything. This could be even in your personal life. Doesn't matter what. Is there anything right now you're doing that you're really excited about that you?
Oh yeah,
actually,
it's actually iGEM related. So because we are so passionate about our iGEM experience, and we are thinking that there's nothing in this like curriculum that we had is similar to what we do at iGEM. So we're actually trying to we're actually delivering right now a student-directed seminar on biological engineering and biotechnologies. So it's actually being run by me and another previous iGEM member. So right now we are delivering this seminar with 10 other students, and then talk about things in biotechnology and also about synthetic biology, all the emerging field and what has been done before. And then we're also like trying to make an analogy of what we do at iGEM, where each of the person will come up with a project to address problems in the real life using principles in bioengineering. And then that has been truly interesting and also very fun experience, trying to facilitate a seminar, also meet people, and also extending our experience at iGEM to more student bodies at UBC.
That's excellent. All right, and Janella, and you?
Emilia, you absolutely took what I was going to mention. Yeah, I think that's especially useful because what we're doing right now with a student-led seminar, it's only a term long, but it's kind of a primer to be able to turn something like iGEM into something that's credit-based, so you could enroll as a course in it, and instead of it being completely volunteer-based, which it is now, you could actually get some credit for it and have some like standardized and well-documented material that goes down in terms of training and past project and things like that. So I think that's truly great. I think personally just something I've been exploring, I guess, and that I find really fun personally is delving into the world of scientific illustration and things like that. I think there's a couple of great artists or initiatives that like iGEM, where they try to bridge this gap between biology and engineering, then science illustration is kind of the gap between biology and design. And something that also tries to bridge this gap is a competition that we were part of in 2020, which is called Biodesign Competition. And it's similar to iGEM in terms of it is an annual competition with an annual project by universities, but it's more centered around making biology-based design in relation to art. And it has a more design emphasis. And I think that's a really great initiative that if anyone is interested in making a team or joining, I'm not sure if there's any like a UBC team for it this year. I think that's something great that emphasizes that interface between biology and design, which is really fun.
Absolutely. Absolutely. I want people to get involved in that too. One of the things we talked about, you both so wonderfully said, is the advantages of interdisciplinary science, right? The more barriers you remove between people, the more we realize like, oh, you can help me and you can help me. I mean, the both of you can literally just go to the next lab bench over and be like, hey, Janella, can you help me out with something? You know what I mean? So I think that's important, but I also think bringing in art and bringing in expression and bringing communication into that space as well is deeply, deeply important. That's me just ranting now. So anyway, thank you so much, the both of you, for your time. It is so exciting to learn about both of you and what you're doing. And you are obviously going to make dents in the universe, and I can't wait to see what those look like. So thank you very much.
Thank you so much.
Thank you. Thank you for
it was a really
us.
fun experience. Thank you so much for inviting us.